Good morning students, this is Dr. AMRESH'S from AMRESH'S classes. Today we are going to discuss about what is cell as well as mostly we will find what are the different organelles present, what are the structure and its function of a plant cell. Okay. As we know earlier that what a cell is, Robert Hooke is the scientist first discovered the dead cell from the bark of a tree, and he has written a book known as Micrographia, and he has discovered one microscope named phase contrast microscope. In that he has shown that the cell's structure is actually resembles with the beehive, the honeycomb-like structure, and that was the dead cell. The latter 1723 Antony van Leeuwenhoek has discovered the living cell from the RBC of man. So let's start today. The plant cell will start about what are the ultrastructure of a plant cell is. So from the external morphology, it is clear that it is rectangular in shape. It is little bit more rectangular in shape, and here it's having outer covering which is triple layered, outer covering. You can see this is upper layer, this is lower layer, and is the middle layer. Okay, upper layer, lower layer, and the middle layer. Upper layer is ectoplasm, lower layer is endoplasm, and the middle one is middle lamella. Okay, so out of all this, we can say if the fruit is, we know that the fruit is it's a ripe fruit. How the raw fruit is converted into ripe fruit, you know, because this middle lamella, it is ruptured or destroyed, then the raw fruit is converted into ripe fruit. So this is the middle lamella. Okay. After that, just you see the structure it will find out here. The outer covering is known as cell wall. This outer covering is known as cell wall. The cell wall is made up of cellulose, hemicellulose, pectin. Cell wall is made up of cellulose, hemicellulose, pectin, lignin, suberin, like this deposition of the material that composed above this cell wall. Actually, total cell is divided into three distinct parts. One is cell wall or the plasma membrane, second is cytoplasm, third is nucleus. Here it is cell wall, cytoplasm, nucleus. Total the cell is divided into three distinct parts. One is cell wall. Here the first one, upper one is the cell wall, and inside the cell wall, the total cell inclusion is suspended under a fluid-like substance which is known as cytoplasm. So this total area you can see, this total area is covered with cytoplasm, and inside you will find a bigger spherical dark shaped body which is present inside the total cell, and it's meant for control and coordination of the total cell. It's known as nucleus. Okay, let's start with cell wall. As we know that cell wall is made up of three distinct layers, and it helps for the protection of the cell. It provides the external skeletal support as well as it helps in protection to it provides protection to the cell. Number 2, this total area which is present here inside, this is called as cytoplasm. And almost all cell organelles which are present inside, they are always in the floating mode in the cytoplasm. So here in the cytoplasm, what are the cell organelles? Let's discuss one by one. First of all, we'll see inside this ribosome is there. Okay. Ribosome. So if you'll find the structure of this ribosome, you can find this here. This is ribosome here. In this area, it is ribosome. And here in this ribosome, we'll find three distinct structures. They are always in two subunits. Ribosomes are three types. One is, here you will find (50-30). This is 70s. This is 80s. A stands for spread box unit. Okay. Another in the lower organisms, 55S ribosome is also available. So these are the ribosomes having two subunits. In case of 70S, 50 is the bigger one, 30 is the smaller subunit. In 80S, 60 is bigger one, 40 is the smaller subunit. So total 70S and 80S. S stands for the spread box unit, the unit that measures the ribosome quality. And what are the function of this ribosome? It helps in protein synthesis. It helps in protein synthesis. Next to ribosome, another here we will find these structures. This is known as endoplasmic reticulum. Endoplasmic reticulum. You can see here and here. Where this location actually? This is nucleus, and from the nucleus they will start extending, and it will extend up to the cell wall or the cell membrane. This endoplasmic reticulum is there, and this endoplasmic reticulum actually two types. One is smooth endoplasmic reticulum, another is rough endoplasmic reticulum. Here, smooth endoplasmic reticulum, actually no ribosomes are there. That's why it helps to increase the area of absorption. And rough endoplasmic reticulum, as here rough, the word meant for the ribosome is present and helps in protein synthesis. Ribosome present. So here these are endoplasmic reticulum and these are ribosomes present in rough. But in smooth there is only lamellar structure, no ribosome is present, as this endoplasmic reticulum divides into cisternae, vesicles and vacuoles. Okay. After this ribosome, the endoplasmic reticulum, another cell organelle you will find, that is called as mitochondria. What is that? Is mitochondria. So this is the structure of mitochondria which is present here, and this is the external structure of the mitochondria. And here what we find, this is the outer membrane, this is the inner membrane, this is outer chamber, this is inner chamber, and here the dotted cell particle is called matrix. Okay, what do you find here? That is the total structure. You will see this is two-layered envelope is there in mitochondria. This is outer membrane and this is inner membrane. Outer membrane and inner membrane, the space is there. This is perimitochondrial space. What it is called? Perimitochondrial space. And you see the inner membrane is folded into the finger-like projections. Like these finger-like projections are there, the inner membrane is folded into. Okay, that is called as cristae. These finger-like projections are known as cristae. So you find here the mitochondria as the total inside of this, the dotted cell structures, which is the fluid-like structure, that is called as matrix. And inside the matrix, the mitochondrial inclusions will always in the floating and cyclosis condition. Okay, and here we find the DNA, as mitochondria is having its own DNA, ribosome, and here the dotted cell particles you can see here around these finger-like projections of the crista. This is known as F1, F0 particles, Fernandez-Moran particles, or F1 particles. Like that we can say. So these particles are mostly helping storing this energy in the form of ATP, adenosine triphosphate. So by storing this energy in the form of ATP, the mitochondria is known as powerhouse of the cell. Mitochondria is known as powerhouse of the cell. But what's the ATP is called? ATP is called as the energy currency of life by Liebman 1940. So ATP is called as the energy currency of life, was proposed is Liebman 1940. Okay. So you know about this mitochondria. Then we'll find about this bigger sized one, big sized one. What is this? And this is vacuole. So here we will find three different types of vacuoles. What are they? What is vacuole then? You can see the biggest, the swelling like structures or the bulging like structures or the big style structure which is present inside the cell of the plant. It is known as vacuole. This outside covering you can see here. This outside covering, this is vacuole. Okay. This outside covering you can see. This is called tonoplast. What is this? Tonoplast. The outside covering which covers this vacuole is known as tonoplast. So vacuoles are generally three different types. What are they? Food vacuole, then water vacuole, this is water vacuole, food vacuole mostly found in amoeba-like organisms, and contractile vacuole that are also found in amoeba. Okay, the vacuoles are how many? Three different types. One is water vacuole, second is food vacuole, third is contractile vacuole. Water vacuoles is found in everywhere, almost all plant cells. Food vacuole is found in lower organisms, example amoeba, and contractile vacuole is found in exclusively amoeba. So we will discuss about these vacuoles. So what the vacuole meant for? First of all, these vacuole, which are the water vacuole, here is maintain, regulates what? The excess amount of the water if will come inside, then it can maintain and regulate its body to expel it out. Food vacuole, when the amoeba takes the food inside, then food is reserved inside this vacuole known as food vacuole. Contractile vacuole, in case of amoeba, when amoeba receives much amount of water, then it will swells its size. That is diastolic size. That size of the amoeba when it receives water, absorbs water by the process of diffusion, it swells up. That shape is known as diastolic size. And the size will be maintained in the body by this contractile vacuole. Okay. After this vacuole, then we will find about different structures like this. What is this? This is chloroplast. Okay, so this is called thylakoid. This is lamella, stotalium, grana, this dot shape structure, stroma. Okay, this is outer membrane, this is inner membrane. So it will study the structure of plastid. Plastid, actually 3 types are there. Moreover, distinctly it is divided into 2 types. One is colored plastid, one is colorless plastid. But moreover, if you will see, if you will search about, if you will find out its characteristics, its work, its function, then 3 different types are there. One is chlorophyll, chromophil, or you can say the chloroplast, chromoplast, and leucoplast. Okay, leucoplast are colorless plastids, chlorophyll are only green color plastids, and chromophil are found in orchid plants. So these are colored plastids. Okay, so let's start with chloroplast. What the chloroplast is made for? You can see it's the pile of coin. You know coin. The coin, if you one by one, if you keep this coin with a bunch or bundle of the pile of coin will be there. One single coin is known as thylakoid, and the bundle of coins are there one after another. If there, then that is pile of coin, and the piles of coin is called stock of thylakoid, or it's known as grana. This total is grana, and grana is connected with this grana lamellae, and here the dotted structures which are viscous fluid are there, white in color or colorless. It is known as stroma. So in case of photosynthesis, grana meant for light reaction of photosynthesis, while stroma meant for dark reaction of photosynthesis. And the photosynthesis process, you know, it will take 4 elements like chlorophyll, sunlight, carbon dioxide, and water to form carbohydrate type of food or glucose type of food by the process of photosynthesis. Okay, that's why green plants are known as autotrophs. So this is all about your chloroplast helps in photosynthesis. Next, after all, you will find here microtubules are there. Here the microfilaments are there. These microtubules and microfilaments, these are having important role during cell division. Then you will find here another substructure is there. Here, what is this? Now this is Golgi body, Golgi apparatus, gamma cytomembrane, or in case of plant cell, this Golgi body is known as dictyosomes. What it is known as? Dictyosomes in plant cell. If you study the structure, you find here, you can see here, you find this is one phase, this is other phase. This is maturating phase or maturing phase, This is forming phase. Okay. And this Golgi body made for secretion of different hormones. Okay. The acrosome secretion. You know the acrosome? In case of sperm, the acrosome is there. The acrosome secretion is also by the Golgi body. Different gastric juices secretion, hormone secretion is also by this Golgi body. So Golgi body meant for only secretion. After that, another something is there. You can see mitochondria, Golgi complex, chloroplast, grana, mitochondria, all this stuff. So now comes about the most important organ, most important cell organelle in cell. Is that? That is, here you can see this is nucleus. It is spherically dark set, present centrally located and helps in, what is that? What's the function of it? It's control and coordination of the total cell. It maintains the control and coordination of the total. You can find the envelope. You can see here the nucleus is covered with the two distinct layered envelope, two distinct envelope layer here. And inside that a spherical structure is there which is known as nucleolus. It contains mostly RNA, helps in protein synthesis. But nucleus inside you will find chromosome. Chromosome, these are the chromatins. Two chromatins you will find. One is euchromatin, one is heterochromatins. These chromatins are meant for what? This will transmit the characters from one generation to the other. These chromosomes. Because inside the cell we will find nucleus. Inside the nucleus we will find chromosome. And inside the chromosome we will find DNA. And inside the DNA we will find gene. So genes are the regulator by which the characters of parents will transmit to the next generation, to the son or daughter through these genes only. That's why the whole control and coordination is controlled by this nucleus. So nucleus plays an important role here. So students, today we have discussed everything about ultrastructure of a plant cell. Okay, I hope you will understand well. Thank you. Bye bye.